Is the screen visible?
Yes.
Because I can see here you are screen sharing. I can see you.
Mm-hmm.
And I can see Abhishek's very beautiful flowers.
Mm-hmm. Okay. So Facebook Live is starting.
Okay.
Um Abhishek, you can proceed, please.
Yes. Good evening, everyone, and welcome to the third day of our wonderful 5-day lecture series in celebration of the World Ayurveda Day. On behalf of Council of Ayurveda Research and on behalf of Ayurvidya Anusandhan Abhiyan Foundation, I'd like to welcome you all. As you all know, the CAR and AAAF are a complete are organizations which are started by Dr. Pratibha ma'am, and it was started with the vision to promote and establish Ayurveda as evidence-based health science globally. Our mission is to encourage, educate, and facilitate basic clinical and interdisciplinary research in Ayurveda. We are open to donors, and I would request you all to become either a founding donor or to become a member donor of CAR and AAAF respectively. So it gives me immense pleasure to introduce today's speaker. So today's speaker is Dr. Asmita Wele ma'am, who requires no introduction. She is a professor and head of Department of Vasarishta and Bhaisajya Vigyana Kalpana at the College of Ayurveda at Bharati Vidyapeeth University in Pune. She is also the honorary professor at the University of Debrecen, Hungary. Her expertise lies in designing and executing of end-to-end research projects of Ayurvedic formulations. Apart from standard formulations of asava aristas, telas, and herbal minerals, she has experience in making parpati, sindura, potli, abraka sattva, etc. She has been the principal investigator in a lot of Ayush projects and a member of the Ayush International Cooperation Committee of Scientific Advisor Committee of Gujarat Ayurveda University, Jamnagar. She has guided about 15 MD students and 2 PhD students and has published over 40 research papers, articles, chapters, and has also authored a book on research methodology. She is a reviewer of some indexed journals. I can go on and on, but I want to say a few more points. She is an Ayurvedic expert for the historic working group meeting for the development of WHO benchmark documents of Panchakarma, Ayurveda practice and training. She is an active member of many organizations. She has traveled extensively to Europe, to USA, Mexico, Africa, Middle East, UK, etc., on professional assignments. And consistent with her family legacy of Vaidya traditions from Goa, Dr. Asmita has earned her BAMS and MD degree from the University of Pune. She is also a practicing physician of integrative lifestyle management for cancer, respiratory diseases, sleep disorders, and PCOD. And on this wonderful day, or for the celebration of Dhanvantri Day, I would take this opportunity to invite Dr. Asmita ma'am. Over to you, ma'am.
Thank you, Abhishek, for that elaborate introduction. I mean, I was also listening intently. What's going on, right?
I can go on and on speaking about you, ma'am. It's an
Namaste,
honor to have you,
namaste.
ma'am.
Good evening, everybody, and hello, Pratibha. Um, I'm not sure whether it's too early for you in the morning.
No, it's not.
Okay, and you are up and awake. So, this I must take this opportunity to say that Dr. Pratibha and her team at CAR USA are really taking, fathomless efforts to promote Ayurveda in a very meaningful and scientific way. They all are working day and night, and they are not leaving a single opportunity where they can, put and promote Ayurveda on the mainstream pedestal. So CAR and AAAF, congratulate you, and I wish a lot of success to you and your team, especially on the eve of Dhanvantari Day today. This has been a wonderful and meaningful attempt of Ministry of Ayush, again, to focus and bring forth the best of Ayurveda, not only for the Indian continent, Indian subcontinent, but for the whole world. And that's why the vision of this, or the mission statement for this year's Ayurveda Day, on the background of the pandemic COVID-19, is how Ayurvedic people are seeing Ayurveda in 2030. How we can bring up different ideas coming from different biosciences and Ayurveda together for the benefit of mankind. And I'm really fortunate to have been invited by Kar here to talk to you. I know that two of my colleagues and friends, senior friend Dr. Lakhotia also, they have spoken on integrating, or how to bridge, or how to bridge the gap between Ayurveda and the conventional sciences. And two of my colleagues will be talking to you tomorrow and day after. So today I'm in between, and I'm trying to and I'll be talking to you about how and why scientific writing is very essential for global outreach of Ayurvedic research. So in Ayurveda, people are working, they are doing research. Also, there are many people out there who are out of India at the moment. They are in Australia, they are in Germany, Hungary, France, even in Argentina, Chile. A few of my friends are in South Africa. So many of the friends are in USA, and all of them, many of my students are in USA. Many of my students are in some parts of Europe, and everybody wants to do something um, focused for Ayurveda. My question or my request to all of them is, since you are working there, you are working in a different atmosphere. You are working where Ayurveda is not regularized as a system of medicine. We fall in the category of complementary alternative medicine or traditional medicine, as the country focuses. And there, you are practicing, you are giving therapies, you are giving advices. So it would be really nice for you to write. And the response is, yes, we want to write, we want to do, but our papers are getting rejected. Many a times we submit projects, but they do not get acceptance. They do not reach to the level of funding. And that's where I started looking into this area, that, okay, we will be doing very good research using different modern methodologies which are available. But what if the research doesn't get published? And then what is to be done if we want to get all the things what we do? We have to write and we have to publish. So that's why I present these thoughts to you, scientific writing for uh, global outreach of Ayurvedic research. The present status of Ayurvedic publications is like this. Somewhat I searched, glanced through various sources, and then these are the facts which I want to put in front of you. Who publishes? Non-Ayurvedic people who are very much interested in Ayurveda, who are from pharma industry, who are from biology. There are a few from modern medicine, Western biomedicine also. There are very many botanists and herbalists. So they want to publish. So that's one category of people from India as well as from abroad I have seen. Then I noticed Vaidya scientists. This category Vaidya scientist is for MD, PhD Ayurveda students. They will become Ayurveda practitioners, but at the same time, it is expected that they take the role of scientists. They research, especially those who complete their PhDs, and they publish. And then rest of the Ayurvedic fraternity, many physicians who after practice about 10-15 years, they start talking about different difficult cases which they have handled. They have tried to cure the diseases. They have tried to maintain the health. They have tried to maintain that feeling of well-being in the patients, and they talk on different platforms about their cases. Most of the times these cases are they do everything, the patients, and as a last resort they turn to Ayurveda. And these are chronic psoriasis type of cases, chronic amla pitta, and whatnot. There are many more. So those, but those patients get relief and their symptoms completely disappear. And then these people, they also write and present. Then coming to very recent, in last 5 years, what I have observed is about 3,000 postgraduate students take admission per year, and the publication number is just 200. There are standard journals and there are substandard journals. These publications appear either in standard or in substandard journals. The ratio of substandard journals to standard journals is 80 to 20. So out of these 200 publications, only 20 publications come in standard journals, which are listed in the Scopus, PubMed, or Web of Science databases, and such others. We can see that the number of publications has definitely increased. It is due to some mandatory requirements of completion of that master's degree or the PhD, and therefore there is a lot of scope of improvement. Most of the times the publications are kind of repetition of compilation and translations of various informations. I also could figure out lack of awareness about recent works on the topic from other branches or branches other than Ayurveda. Also, people working from different countries. Recently, I noticed a thesis on honey. I was a reviewer of that work. The student has from Australia has worked on medicated or medicinal honey, and that information was really brilliant. Lots of work has been conducted on honey around the globe, and I could see all that information at one place in that one thesis, and it was focused. But if I coincidentally simultaneously I had to check one thesis of a renowned university here on a similar topic of honey, and to my disappointment, the information coming from Indian Ayurveda sector was so limited only to the traditional ancient texts. So what's so that talks to us about the gap which is there in the system, in the Ayurvedic fraternity, here and outside also. I think I also could figure out that the root cause of such a difference is the attitude, which we can see, which is palpable from the UG and PG education, where students just recite the textbooks. They study for the sole purpose of passing out, getting degree, and then a license to become a doctor. So that's what is responsible. So all these facts, that is responsible for the present status of Ayurvedic publications. With this, I also went a little deeper and I introspect. I talked to many of my students and I could figure out these reasons why there is such an apathy to write and publish. Most of the times, even the physicians who are working, practicing in the field for 20, 25, 30 years, they find it difficult where to start. They ask me that, yes, we don't understand how to start, where to start. Then there is anxiety about writing skill. Ayurveda is in Sanskrit and it is taught in regional languages. So there is always a little bit of apprehension about the writing skills, which leads to lack of confidence. And then uh, in the modern world, where we always need to translate, the knowledge is transpired between two different systems, pathies, two ethnicities, different language groups, and this translation is a difficult or deterring factor for anyone who wants to write. To satisfy the scientific community on both sides, Ayurvedic community, we have very nice very sound philosophical foundational principles. We have very strict methodologies. We have application parameters. We have testing parameters. So to fit our work into our parameters and also make it, convert it in the known sciences parameters, that becomes a very difficult jargon for people, and then they think that, okay, should I write, should I not write, and then they find local platforms, Vaidya groups, and then magazines and other areas where they write, and it is very welcome that they should write there. But here we are talking of scientific publications, and this is one of the reasons they cannot write, or there is little apathy towards writing. By overcoming all these obstacles, if somebody writes, then there is a fear of rejection, and post rejection there is inertia. Every scientist who writes and who publishes understands these two phenomena. First of all, I write something, my student works very hard for 2 or 3 years, for MD, 4 years for PhD, and then they are writing, and then I'm a co-author or I'm their supervisor. They write, and then when it comes to various journals, we get rejections for 3 or 4 journals. There are different reasons. Even I write a paper today, I get rejected. And then this post rejection inertia sets in. Why should I write? No, I know whatever I wanted to say, I have said it. Then why should I do it again? So that's another reason for less publication number from Ayurveda. So let me tell you that science is all about experiences and experimentation. Writing is expression of emotions and experiences both. So scientific writing is acquired skill to share the knowledge gained through experience and experiments. And for us, the fraternity of Ayurveda, it is very, very essential to acquire this skill. Once we accept that what we write is not the writing of a poet, or is not the artistic creativity of an artist, it is a scientific technical skill, then we can learn it. And with this approach, we will see how to write, what to write. This scientific expression, where, at which places we need to communicate, we need to write. So generally, this scientific writings or the scientific communications happens at 2-3 places. First is oral communication, right? I'm talking to you now, I'm giving a lecture. You talk in conferences, you talk in seminars, you give seminars, you talk in workshops, and also you demonstrate along with that. Then you conduct focus group discussions, you conduct panels. All this is oral communication. Then there are written communications. You write MD dissertation, PhD thesis, research reports and projects. You write original paper. There are different patterns of writing, and a rule set or conventions for writing of each type is different. So you write original paper, you write short communication, you write a review paper, you prepare a poster for presentation in a conference. There are PowerPoint presentations when you talk from the dais along with your oral communication. That is also writing. In various journals, there are discussion kernels. There are letters to editors and short and other types of communications. So for all of this, we need to write. We need to write proposals for funding agencies. We need to write applications to approval committees like ethics committee. If you are conducting animal experimentation, if you are conducting a clinical research where human participation is involved, you need to apply in a particular format to the ethics committee. Your research content remains the same, but what you should write for what differs. And you have to focus different aspects of your work according to the need, according to where you are submitting it. For example, for writing for patents, the format is completely different than when you write a MD thesis or a dissertation. So these are the platforms where you write, where you communicate. What is the language of the text? a common problem which every student or a scientist goes through. What is a terminology? The terminological words coming from Ayurveda, like dosha, vata, pitta, kapha, like kvat, like paricharya, these are terminological words and they should be kept same. Even if you write your paper in the journal like Nature, I hope someday our paper will definitely appear in such a high quality journal. The terms which are non terminological, like nirman, nirman vidhi of, you do not, I mean, there is no need to, write this word nirman. You can write preparation of kwath or making of kwath. Very simple. Another area where there are scientific terms, in modern science also there are you will find two types of terms. One is uh, scientific terms and lay terms. So science is always translated for layman, and there is another big area of scientific writing for lay people. That's one of the interesting areas. So whatever new research happens, that research is translated in simple languages for lay people. And here, when I'm writing for a journal, I should use the term hyperlipidemia, whereas when I'm writing for a lay person, I have to use the term high cholesterol. So this is the difference between how you should choose your words and sentences when we are writing for a transdisciplinary audience, people who are interested in Ayurveda, but they are not core Ayurvedic people. They are from different biological applied sciences. Some might be chemists, some might be biochemists, some might be botanists, zoologists. So for this diverse but scientific population, it's always better to read the pertinent area to your work and then use the scientific terms which are used in that science. Apart from this, there are I want to draw your attention to two three different types. Like I said, that there are uh, reviews which sometimes you have to write. There are reviews about concept, reviews about disease, reviews about phenomenon. Then people write books and you have to write a book review. Conferences are held and you have to write gist of that conference. What was the theme and what was the take home point? Then there are sometimes you have to write a comment on a report. You have to write a comment on a paper. Or when you are a reviewer of journals, then you have to write reviewer's comment. Nowadays these reviewers comments are published on Web of Science. It's Publons. So these are different types of writing, and the technique, all this is related to science. This is all scientific writing, but the techniques are different. Today we will confine ourselves to writing for scientific paper in the category of experimental and clinical research. So first, I'll just briefly take you through these 4 points. A scientific paper is a written and published report describing original research results. It's a scientific paper. You have to always remember that it must be the first publication of original research results in the form whereby peers of the author can repeat the experiment and test the conclusions. Why it is important, I'll tell you later. And in a journal or other source document readily available to the scientific community. So this particular slide tells you about scientific paper, its originality, its reproducibility, and its easy access. So let's see how to make this happen. For this type of experimental, clinical, observational, descriptive research type, the format which is followed is IMRAD. I stands for introduction, M for methods, R for results, A, it's dicey. Sometimes it's taken as and sometimes it's taken for analysis. and D is for discussion. So in a paper, now this is a past tense. You have worked, your study is complete, your experiment, your clinical trial is over. Now you want to write it. So in introduction, what was the question that was studied? That's written. Then how the problem was studied, how part is covered in methods. What are the findings? That's covered in R, results and analysis. How are we analyzing the results? Which methods we are using for analysis? Are there standards, controls, statistical tests applied? So all that comes in analysis. And then discussion. What do these findings mean to the problem at hand, and how we are going to contribute to the larger body of science? That's what comes in discussion. Okay, here it is Sanskrit. Let me take you to my very, very favorite Samhita, Charakasahita. And in Charakasahita, the same thing has been written in no, in very, very clear terms, no uncertain. So it's very clear. Atha pratidnya, atha sthapana, then pratisthapana, hetu, drishtanta, upanaya, uttara, siddhanta, and all of this together as abhyupagama siddhanta. That means when there is something which is not known till date, like COVID-19, we have to find out. There are no answers today available for this COVID-19 with modern medicine or with us or with any other science. So everybody is working towards it. So that's anukta, apariksha, which is not known till today. That particular thing, when you have to study, it's abhyupagama siddhanta, and this is the methodology. This methodology I have put for your understanding in concurrence. So in modern, we say in present day understanding of paper writing in the IMRAD format, we say that introduction covers the scope, aim, and hypothesis of the study. The hypothesis is alternate and null. So we are unbiased. Aptaha shishta vibuddhaste. So we are unbiased. We want to taste, we want to verify what is going on. Whether this particular set of drugs is effective in COVID-19 or not. So this is the hypothesis and alternate and null hypothesis. Alternate hypothesis generally talks about the positive side. That means this will work. And null hypothesis says that it will not work. So here we talk about pratidnya, which has two parts, sthapana and pratisthapana. So hypothesis, alternate, and pratisthapana, null hypothesis. Then there is materials and methods in the IMRAD format. Here it is called hetu, which is set with the help of pramanas and drishtanta. Drishtanta is example. So the pramanas with examples or experiments, we analyze this. We analyze the whole issue. We analyze, or we see whether the pratidnya is going to be, sthapana is going to be correct or pratisthapana is going to be correct. So that methodological set is hetu. Results and interpretation. Here in Ayurveda, we call it as upanaya. So whatever experimentation we have done with the help of pratyaksha, anuman, upman, and all the drishtanta and everything, we relate it to our sthapana. So whether it is relating or whether it is refuting, that is upanaya nigamana. So it's kind of oha poha. It's proving and disproving. So that's the discussion. Discussion ending with conclusive factual statement is uttara and siddhanta. So siddhanta is after so much of deliberation, after meticulous objective planning, following the method, discussion, deliberation, this is right, this is wrong, this type of result is coming. Pakshata, siddhanta, pratipaksha, all of that. And then you come to a nirnay. So that is siddhanta. And the same thing which you can find in Charakasahita, which we know today as IMRAD format, I am convinced after studying this so much that the methodology or thought process is same. The only difference is of language and experiences through different centuries. Otherwise, this is very, very possible. So let's see what should be there in introduction. What is the problem? What is the important issue today? COVID-19. What is already known about the problem? We know that this is a virus, this is SARS-CoV-2, and then lot of things about it. It is RNA virus, this, that, everything. Which model or methods have been adopted? So there is one way of going ahead with antiviral technique, whether there are various drugs which are already known as antivirals. In Ayurveda, we might think that yes, it is Jwara, and the Jwara which is with Rasava Srotas and Pranama Srotas, and we know the certain medicines, we have certain models and we want to use them. What are the unanswered aspects or gap in the knowledge? So we have to find out, for any problem, we have to find out that yes, there is no answer to this particular small fraction of the big problem. Like for example, in diabetes, in cardiovascular diseases, in psoriasis, we say that yes, many things can be done, yet there are no complete or 100% cure. And that's why the whole science is that's what true is about the whole science. Science is always for inquiry and finding out answers, solutions. Certain answers are not suitable in one situation, certain answers are not suitable in the other situation, and you have to keep on studying. So to study that, what approach you wish to take? All these questions should be answered in the form of statements one by one in introduction, and thereby you put your aim and objective. I'll give you one simple example. Here, example is of pain. Now there is a historical context. Now this is type of introduction. So pain is most common. What we are studying, most common. But is it important? Yes, because it is the fifth vital sign. It also gives us the scientific importance that it is a protective mechanism of the body. Whenever pain is there, your body is telling you to take care. Don't let go. Don't forget. Don't neglect. You have to take care. And then there is economic importance. Because 30 to 50% of the world population, if suffers from pain, that means if you are finding out a drug for pain, it has a lot of economic importance. There are many painkillers available in Western biomedicine, in traditional herbal medicine, in Ayurveda, in homeopathy, in Siddha, everywhere. So what's big deal? There are conventional pain management methods. So you take a critical review. Basic, how many methods are there? Drug methods, non-drug methods, therapies, acupuncture, acupressure, Ayurveda, painkillers, everything. So you take review, and then you come towards novel idea of your mind. Whatever you think that, okay, so much is known, but still there is some novelty to my idea. Then you have to spell out that idea at the end of the introduction. And for that, you take evidence from Ayurveda publications to look for new leads, new formulations. This is how one has to build the introduction. After the introduction, you have to write materials and methods. A detailed writing of materials and methodology, unassumingly. I'm using the word unassumingly because when you are writing something for a transdisciplinary audience, readership, then you have to assume that they don't know anything about what you are talking. So you have to write about the purpose, which helps to replicate the results. Still, it has to be, the methodology has to be very clear, concise, and accurate. So all these terms are, you are going more and more into the tighter and precise areas. For example, coming from Ayurveda, I definitely know that I am working with Rasashastra, Rasaushadhis. I know what is Svedan process. I know what is Maran process. Everything is fine with me. And I assume that yes, people who have studied Ayurveda, they all know it. But when I want to do this test, ANC, acid neutralization capacity test, which is of Kapardika Bhasma, which I conducted on that Bhasma as per USP, United States Pharmacopoeia, like regulation number 23, then I have to spell out all these micro details. How much was the amount of Kapardika? 250 milligram mixed with water, but yes, 70 ml of water. Then I stirred it for 1 minute at 300 RPM. I used magnetic stirrer, not the simple stirrer, not the glass rod stirrer. Then I added one normal HCl. I continued the stirring for 15 minutes. Within exactly 5 minutes, the mixture was titrated. So all these details are very, very specific, because when a person who has purchased this Kapardika Bhasma maybe in Hungary or in Germany or in USA, that person if performs this test, the result should be matching and similar. And that's where replicability comes in. So this is true not only for these kind of lab experiments, but this is true in case of clinical trials also. So my second example is of clinical trial. Here there is a study drug, then there is a comparator drug which is inhalational steroid and oral steroid beclomethasone. Then there are placebos which are similar to both. Then what are we doing in the visit? So there are different levels of visit: screening visit, baseline visit, study visit. You can see this presentation later also, so I'm not going to describe each and every step which is written, but I want to make it a point that you should be writing each and every step here. So baseline visit is there, follow-up visit is also there. I would like to say that when it is a Ayurvedic research, then it's always expected from the Vaidyas that you should write the chronotherapeutics. That means if you are prescribing a medication to your patient, tablet of 125 milligram 3 times, okay, anyone can write that. But you are prescribing it at Rasayan Kal, Vyanakal and Udanakal, and its equivalent meanings: early morning, after lunch, after dinner, or after morning meal, after night meal. So this is how the detailing is expected in methodology. You can see here this small chart where there is visits, where there are visits written V0 to V17, and this is end of the study, that is visit 18. And you can see here that inclusion exclusion criteria happen at visit 0. Treatment starts from visit 1 to visit 12, and then there are some safety laboratory tests which are to be conducted. I mean, in this study they were conducted at V0 as well as at V14, and this is how you can give the methodology so that people can, the next researcher can replicate it. After the detailed methodologies, we will enter into the result section. In different papers, you will find different terms. It depends on the type of work or your area of expertise, what terms you use. So the terms which are used as observation, results and analysis, or interpretation, sometimes results and interpretation, observation and results, results and analysis, only results, these are all used interchangeably depending on the journal's requirement, depending on the area where you are working, and then you can use any of them depending on your requirement. Results constitute half of the main body. So main body is results and discussion. Half of it is results. It is your own work. You have collected the data, you have organized the data, and now you are going to present it. So if you use, along with the text and writing, if you use tables, figures, charts, and diagrams, your results become very, very impressive. For example, I'm sure that all of you are aware about the term, just like doshas, Prakriti. Prakriti is very near and dear to the heart of all Ayurvedic physicians, students, researchers, everybody. We all, because that is the simplest tool in the hands of physician anywhere in the world without any machine. We have this tool of Prakriti and we understand the people, patient, and we can design the treatment pattern. Now, in India, a group of Dr. Mitali Mukherjee along with Bhavana Prasher, they are working very seriously over a long period of time regarding different aspects of Prakriti and methods how we can, how we can incorporate these principles and theories of Prakriti into the mainstream scientific thinking. So this in this paper, in this study, what they have done is they studied a group of uh, Vata Pitta Kapha predominant healthy volunteers on to find out prevalence of their gut microbiome. I hope I'm clear. So we all know that at one level, all the individuals, we have a Prakriti, we have certain things in common, and at the same time, every individual is different, that is heterogeneous. So genotypical and phenotypical expressions, they vary in individuals, yet there is a commonality, there is a common basis. And this group, they studied in a very consistent group of people, their gut microbiota. They wanted to ascertain whether the microbiota from Vata Prakriti people, Vata predominant, that microbiota from Pitta predominant, and the same microbiota from the Kapha predominant persons, is it very similar or it differs? And they started this study with this objective. They studied it with 16 srRNA gene sequencing technique was used here. And you can see this graph plotted, p values you can see. The pink dots, that pink is of Kapha, green is Pitta, and lighter shade, lighter orangish is Vata. So in this study, they could differentiate that yes, in the population where diet, environment, age, and the other factors are almost similar, in that population, there was a basic similarity between the gut microflora, but depending on the Prakriti, presence of these these microbes like Bloschia, the new Bacterium rectale, Prevotella, these amounts differ. That means they have found out something regarding some concrete proof regarding the differences between Vata, Pitta, and Kapha predominant individuals using this technique. So, with the presentation, results are very clear. Here is another example. We studied for our Krishna Vajrabhrak for mast cell stabilization in rats, and we wanted to know whether the results of a single dose, you give Krishna Vajrabhrak Bhisma in the dose of 125 milligram once, and you are giving it consistently for 14 days, whether the results change, whether there is effect, whether there is efficacy. With one dose, you check effect. With consistent dosing, you check efficacy. Efficacy is the long-term result which takes us to a punarbhavatva, non-recurrence. So we studied it, and here you can see the results. On the x-axis, there is percentage of unruptured cells. On the y-axis, there is control, there is a treatment group description, and there are two graphs together which are giving us the results. The two stars at the top of the bars, they talk about significance, and the level of significance we have calculated is p is less than 0.001. So this particular one graph tells us complete details without even text. And this meticulousness is expected. When we say that our papers are published in substandard journals, and that's why the world out there doesn't take cognizance of our serious and good work, good intentional work, is because of our poor presentation of results. So the presentation in the past slide and this slide it's just exemplary. There can be many more factors here, but in this presentation we have missed on nothing. We have demonstrated x-axis, y-axis, all these graphs, the treatments first, second, and everything. So this detailing regarding results is very, very important. We will come to the discussion and conclusion, that is in the IMRAD format. There is this last point. In the format, the certain terms are not mentioned, but we have to understand that yes, it is included. We have to presume that yes, discussion comes with conclusion. Again, there are some key points. Interpretation of the data in the context of objectives should be there. We should not move away from the focus. We have a lot of information regarding Ayurveda, and we have a lot of information on the topic of our interest on which we have studied. So generally while writing, we see that we tend to say that Ayurveda is a science of life, 5,000-year-old living tradition, and we come to the disease, and then we start talking. So here, interpretation of data directly, you should start from your own point, and that's what is expected in the context of objective. It's not only that in the discussion you have to talk and focus only about what you have achieved. It's never enough. What you have achieved, you have shown in result and analysis. Now, what you have to do is you have to explain your results with reference to results in other studies previous to you. So you have to corroborate the references of other studies which are supporting your results, and you have to also take cognizance of certain results of other studies which are inconsistent with your result. So you have to accept and you have to refute both, and that's what the term discussions indicate. After that, you also have to critically explain your results, summarize your evidence for each of the conclusion, and your conclusions have to be in tune with the objective. In discussion, the last and most important part is you should be able to jot down the limitations and future work possibilities. I'm going to give you one more example here. There are 2-3 studies. This one is a laboratory study where we did analyze a particular formulation, Vasarishta, from which we made in our lab. And the reason for taking up this study was there were deaths reported after consumption of Mrita Sanjeevani Sura, which is used as a base drug to make Vasarishta. So first you have to make the Mrita Sanjeevani Sura, and then you have to add Vasa juice, Adhatoda Vasika, plant's juice into it. You have to keep it for a while, and then your Vasarishta is ready, which is one of the wonderful drugs for the respiratory diseases. Especially, it's a drug for emergency. And when there is a ban on the base drug, how the other drug can be made? So then pharmaceutical industry, they start finding out their way. So we wanted to study this problem. We followed exactly the text method, and these are the results. So the lab made MS percentage of furfural. Mrita Sanjeevani Sura was made in our lab, and furfural is the toxic content. Its percentage was 1.6 as against the 29.58 in the market sample. See the difference. Furfural is a toxic content. It was present in our sample, but see the percentage 1.6, and then 29.58 was the market sample's percentage. After that, we prepared Vasarishta using this same MS, same Mrita Sanjeevani Sura, where our furfural percentage went down to 0, and in the market sample, it was 149.24. So there was some problem with the process. And this we could identify only with this, and that we have put in our discussion. This explains the dates reported after consumption of this, of the market sample in those days. This was a long backstory. But yes, this is how we have to take our discussion to its logical end. We have to try to present the principles, relationships, and generalizations shown by results in the discussions. And we have to also discuss theoretical implications of our work and possible practical implications. Here was another study. Hingvadi Ghruta and Vachadi Ghruta, these are the two Ghruta formulations which are recommended in Charakasahita and Ashtanga Hridaya for Unmad Apasmar. In Unmad and Apasmar Chikitsa, we'll find these two. We studied these two formulations in mild to moderate depression patients. Now, after that study, since we are talking about discussion, again there was a basic mind, there was a huge discussion how to put the two things together, Unmad Apasmar and depression and psychosis, anxiety. So that was all, the part was clarified in the methodology, and then what we came to know, we have put it in the discussion. So adequate amount of serotonin helps to regulate sleep, appetite, mood, and it inhibits pain. This is what is the pratyaksha, which we can study and find in the laboratory. We also know that some of the depressed people have reduced serotonin transmission. Now, depletion of neurotransmitters like noradrenaline and serotonin. Serotonin means 5-hydroxytryptamine. They lead to the development of depression-like state. I'm giving you the sequence of events. And antidepressants used in the treatment block the mechanism of enzymes and amines, which allow the release of neurotransmitters. That means noradrenaline and serotonin. And thereby, you reduce the depression. And that's how any drug works in depression. Now we have two drugs, Hingvadi and Vachadi. So after studying and after analyzing all these results, we came to the conclusion that Hingvadi might be helping either to increase serotonin transmission or reduce the depletion of the neurotransmitter, whereas Vachadi might be having a completely different action on different neurotransmitters, that means other than noradrenaline and serotonin. So these two formulations have two different areas of work, which we know from our Samhitas. We know that both should be used in different types of disease manifestation. We could give a practical example, or we could give evidence to these textual facts through these two researches, and a comparison between the two. So this is what I mean by discussion and coming to a logical conclusion. Here are some papers, which the very famous paper is from Dr. Forst. There is another paper from molasitosis, one more paper by Gupte, Daune and others. And there is a paper from Patel M, that is Dr. Christian Kessler and Professor Gupte. I would suggest you to find out these papers, which are available in Google Scholar, and you can read it. The last one is not, we have to purchase that. But all these papers are very good examples of IMRAD format. How to write, how to experiment, and how to put the things across for diverse type of readers is very well explained in all these papers. There are many papers out there. I'm just giving these 3-4 examples to, just to begin with. In this IMRAD format, certain things are taken for granted. Title, nobody talks about the title in the format. Remember that title reaches to thousands of readers. So your title needs to be catchy. Once your title is catchy and somebody, okay, is it about depression and ghee? Okay, let me read. People read abstract. So abstract reaches to about hundreds of readers. Then there are keywords. In every scientific publication, you have to write keywords, and there are guidelines of PubMed or Medline and various journals. Certain journals tell you to select the keywords from their list, and certain journals, they want you to give your own keywords. But while selecting keywords, you have to remember that these go for indexing and coding. They are made available via search engines to potential readers. So if you mention some key terms like Ayurveda, like hydroalcoholic fermentation, like Prakriti, like rheumatoid arthritis, like ATP, or maybe when you are doing characterization studies, maybe NMR, then your papers, they reach to the readers. They are coded properly by the coders, and then many readers, potential readers, can access to them. Figures and schematic diagrams are checked by limited number of readers, but these readers are very, very important to you, because they are the ones who understand your concept, they build upon your concept, your research work, and they cite you. So the more the citations you get in your paper, for your paper or your work, that means your standing in the scientific fraternity increases. And all this is related to the H index, I index. I'm not talking on that right now, but I'm just giving you this tip that yes, figures and diagrams are important. So you have to do them meticulously. Full paper is read by only those who are core scientists and who want to work in the area. You have a common mutual area, and there they will read your full paper. So in that sense, there is order of writing the manuscripts. You know, when I was a beginner in this field, I started writing from the introduction. First, I was thinking on a title that, okay, I will be working on this topic. This is my title. And how the way I started maturing in the field of scientific writing, this results and analysis is the basis of research. So first you have to look through the jargon of your results. You have to go for a rigorous analysis. Once your result and analysis is ready, you start writing with materials and methods. So the first part of your research paper is materials and methodology. Then you write result and analysis. Then you bring the essence into discussion conclusion. After this, you write introduction. Because this is a research paper, you have a limitation of words, about 6,000 words. And the patience of the reader is maximum about 6,000 to 8,000 words they can read. So howsoever you love your baby, you have to, make that baby very smart. And therefore, you have to write crisp, short introduction after that discussion conclusion. Then you come to abstract, which is just 200 to 300 words. And remember, 80% of our readership reads abstract. Then go to title, which should convey a complete meaning of your work, but it also needs to be catchy, smart, but not like Abhra Kamahani Bandha. It has to give the essence. Then you come to acknowledgement, author contributions. A research paper is always a teamwork. Even at MD level, even at PhD level, or for any research project, you are working with a team. And the more the authors, it gives out the message without actually writing or saying that you are taking a team with you, you are working with people, and you are a resourceful person, and many brains are put together towards this idea. So that's the importance of author contributions. People are working with you, they are contributing in writing, and therefore the value of the paper increases. And in the end, there needs to be references and bibliography. So this is the order. I have shown this slide just for a second because you need to understand reference styles, and for that you have to use certain reference management softwares. At the moment, the in thing from Mendeley and EndNote is cite while you write. When I'm writing something and I know that, okay, this X person has worked on the area in which right now I'm writing, I can take out, I know that paper, I have read it, I take out that paper and I have to put it, quote it, I just put that reference, I put that paper there, and it gets cited automatically. You get a bibliography in the end. So that is very important. References from texts, there is a ICMJE guideline. It's a group of publishers, and they have, for medical journal editors, this is a group of this is International Committee for Medical Journal Editors, and they have prepared guidelines how to quote the text, where Sanskrit is also included. So how to write a reference from a Sanskrit text, how to write reference from a book, how to write a reference from a journal, all these guidelines are there, and they change, they vary a little bit from each other according to the journal, and you can follow that. Towards the end, I'm going to show you a few tips. See, with my experience, I can definitely tell you that poor experimentation cannot be masked by brilliant writing. But poor writing can mask a brilliant experimentation. So you have to be doing both. Plan a brilliant experimentation and also write good. Take help. That's what teams are for. Someone is very good with handwork and someone is very good at writing, and they have to come together for Ayurveda to write and to experiment both. In the writing, always avoid complex sentence structure. Try to be very simple. Use clear and simple English. There are many softwares which help in correcting English, like Grammarly is there. And in the good publishing houses, language helps are also available on payment. So you can think about that. Always keep in mind that paragraph is essential unit of thought. I have seen a few students who write each sentence in paragraph fashion, and a few students who write pages after pages and there is no gap, no break, no paragraph. So there are different thoughts and there is a group of thought. So paragraph indicates a thought, change in thought, and you have to always keep that in mind. Figures, graphs, tables, charts explain the content clearly, and therefore they must be a part of your paper. Another very critical, crucial issue in the minds of various physicians and also the also researchers. I have registered my title of my thesis like this in the university. I am working with this group of patients. So the title of my paper should be the same or different. Always remember, the title of your paper depends on your results and discussion. What is that particular objective you want to focus, and depending on that, the title is decided. It may not be same as your thesis title or the protocol title or the report title. If you see the Cambrella report, it is a bunch of about 8 different papers under one big project, and each paper talks about a particular factor. So the title is different than that. Ultimately, it all comes under one report, but the papers bear different titles. It's also possible that complete thesis or a project may not be presented in one paper. There are multiple factors. Like we did for our Krishna Vajrabhrak Bhasma, we did Gnasi, we did experimental studies, we did toxicity studies, we did clinical trial, and it's not possible to publish everything in one paper. It is a big volume. So you can divide the work into fragments. But always remember that if you are publishing toxicity study, then it should not be the one part in this journal and other part in that journal. No, the whole concept should come in one paper. Do not invite questions by giving incomplete information. That's very crucial. We assume certain things and we write incomplete statements. And therefore, the information becomes incomplete and the reviewers start asking questions to you. Do not overlook reference style with publishing journal. I just talked about it, and start using reference management systems. So, what makes a good research paper? Good science. good writing in the IMRAD format, and publication in good journal having index factor, SNAP, SJR, and all these indexing scientometrics complied journal. So that's what constitutes a good paper. Sir William Osler once said that in science, the credit goes to the man who convinces the world, not to the man to whom the idea first occurs. So, Eureka, Eureka was Archimedes, but he took the pain to complete that theorem, and therefore the credit is his. In our world, we have to be careful that we should be taking our that small idea to its logical end in a particular format, in a very meticulous scientific experimental methodical way, and then we will definitely take the credit. In the end, I would like to say that when we are aiming at, making Ayurveda a substantial global player in all walks of life, in treatments, in products, in analyzing, in wellness, health, everywhere, people today look at Ayurveda with hope. And if we want to make that, this global scientific community looks at us with two approaches. Either they are skeptical or they are curious. And if we write and publish in the IMRAD format, it takes care of both. Because it is very critical, very neat, with figures, charts, clear objective, clear conclusion, limitations, everything, it satisfies the skeptic, critic. And those who are curious, the paper written in this format also satisfies the curiosity of the curious person. So, descriptive researches, observational researches, epidemiological, experimental, or clinical trials, all of these, they follow this IMRAD format, which is very crucial. And the second important fact, one of my colleague pointed these things to me, that ma'am, if we can write, if all the Ayurvedic research, if that can come in the IMRAD format, then the ratio of standard to substandard journals will definitely go down. That means the standard of journals will come up, because we are publishing a very good research in a very good format, which is acceptable by all. So that substandardness will automatically be filled up. And that's why it's very important to write. I hope I have been I am successful in clarifying many aspects of what to write, how to write, who to write. And yet, if you have some questions, I'm available on my email and you can contact me anytime. So thank you, Kar. Thank you, AAAF India, for giving me this opportunity. And I wish you all a very prosperous Dhanvantari Jayanti and Diwali both. Thanks a lot. If you have some questions, I'm right here.
Thank you so much, ma'am, for a wonderful session by you. And I remember talking to you on Vajrabharat trial when you were in Bharati Vidyapit. And this year is very special because we are meeting virtually more than the physical meetings. And you are like a compendia. Whenever I meet you or I hear you, I learn something new. So thank you so much, ma'am, for being here. And your presentation was like a platter, because you started from a hardcore research trial and simplified the concepts of publication as well as the writing approach in Ayurvedic research. That was very much crucial when we are talking about international publications as well. So I believe that this was useful presentation for all the sections of researchers, right from beginner to the matured researchers. Thank you so much for your encouraging as well as inspiring talk, as always. And it was lovely listening to you again. And I'm sure there are no questions because you made sure that there are no loose ends in the presentation and there are no questions coming up. But just one small little query,
Sure.
or you can say
Sure.
a question from my side, that you mentioned about the ratio, standard to substandard. So can you suggest any checklist where you can highlight first 5 points before thinking that, okay, I need to publish this article? So what are those 5 points which we need to think before submitting it to the manuscript to any journal? Just one little hint.
Sure. Sure. Pranita, this is a very pertinent and right question, that what I should check for whether the journal is a standard journal or not. Going by hearsay, generally all the Ayurvedic graduates, postgraduates, they are aware that there are 3-4 journals.
Yeah.
Ayu is the journal which is in PubMed. Then JAIM, we all know, Journal of Ayurveda and Integrative Medicine, which is also again in the PubMed database. Ancient Science of Life is a journal preferred by many Ayurvedic researchers. That's also is included in the PubMed and Scopus databases. So this is the first thing, that okay, the journal's name which you hear from your peers. The first point is the name which you hear from your peers, seniors, and good scientists in your field. That's the first point. Second point, the journal, there is a list of substandard journals and there is a list of good journals. So UGC CARE list of journals is the list which has been published in May 2019. So in that list, there are various journals. If the journal is included in that list, that means it is a standard journal.
Right.
Now, which criteria these people, this group who has identified predatory journals, they have followed certain criteria. They have checked whether the journal has even if it is new, basically they have followed whether the journal has a legacy. If not, even if it is new, it has proper editorial board. Whether the journal has peer review process. If the peer review process is present, whether it is double blind or triple blind, whether the blinding is there. Then what are the criteria of selection of manuscript? How long the journal takes to identify or to clear the manuscript for publication? Because when an article enters into a peer review process, the minimum time required is 6 weeks. Because at the first level of entry, you submit an article today, you at least take 1 week for the chief editor or associate editor to scrutinize that and pass it on to the reviewers. Reviewers get 2 weeks to check and scrutinize the article properly. After their queries, the article comes back to the author. And then author takes about a week or two to fulfill the queries. Sometimes the data is here, there, you need to add some references, your language is not clear, so you have to make the concept. So it takes about 1 or 2 weeks, and then it is submitted again. At the first level, at the second level, at the third level, all the 3 times generally the article is sent to 2 or 3 reviewers. So the review is received from minimum 3 people, maximum 8 people. And that's where the time is very important. And because of this, the timeline of article publication, which is given in the journal, is very important. Then there are 2 types of journals. One is online, and that means open access journals, where there is a fee for publication. And now, because of the cost of this process, open access good journals, initially there was a phase when good journals never used to charge. The journal articles used to be into the peer review process. Scientific fraternity used to do this work voluntarily, and therefore it was never charged. But now with the online processes, certain expenses have incurred, so there can be a small charge. But then you can see in the list of all these things are very clearly mentioned in the journal website. So you can see the journals, all the details, where in which web databases it is listed. This is the fourth point. So you have to check for the journal, in which databases the journal is listed.
Yeah.
How much is the charge was the third, and then fourth is the databases to which it is attached. And the fifth or the last point to access a good journal is, you know, the scientists with whom you are working, and you know that, okay, they are publishing in these different journals. So for Ayurveda also, let me tell you, you need not confine to these 3-4 journals which I have said. Any journal, the journal from Royal Society of Chemistry group, or journals of ACS, American societies, then annals of medicine, annals, PNAS journals, all types of journals, they can accept Ayurveda paper if you write crisp, precise, with the objectives, with the results, measurable, replicable, simple, and straight.
Yeah.
So then there is no really dearth of good journals. You can submit anywhere.
Yeah. It was a wonderful list. In fact, that should be sticked on the desk of every researcher. It's a very useful list, ma'am. And now we are also having so many good researches which are accepted in other, like you mentioned about non-Ayurveda journals.
Yes,
And we are getting good references from those journals also. And to take it to one step ahead, we are also getting good patents from Ayurvedic researchers. So, and if you are having a good formulation in hand and you think that it is a patentable one, we can have that authority to publish in any journal, you know. So it's very wonderful to have you today, ma'am. And it was very insightful presentation.
Let me correct you a little bit, Pranita.
Yeah.
You have mentioned a very right point, that for patent application, you should publish, but there is also a tricky point. There is a brownie point there.
It should not be
You
public.
you cannot, disclose everything in that research paper. So you should have something published if you are going to file a patent, and you should have something not unpublished for the patent. So
yeah, or maybe in another way, if my formulation is good enough, I should patent it first and disclose everything, and then you can go for a good publication.
That's
so
correct.
Both ways we can have it together. And yeah, it was really useful information, particularly like you mentioned about tips, and that those tips are very crucial and very common mistakes we generally do. So about the paragraphing also, it's very refined comment, and that comes after so much of reviewing also, not only in writing, but when you review so many papers and write-ups by students, you come to know that. It's wonderful learning from your experiences, and I really appreciate your presence today, ma'am. Thank you so very much for being here from CAR, and I also appreciate Dr. Abhishek's presence. He moderated well today's session. Thank you so much, ma'am, for your wonderful session.
Thank you, Abhishek, Pranita, and Meenakshi. I thought she was there for a while in the beginning, and Pratibha also, for being here. And hope to see you sometimes physically, because Pranita, you and me, now we are in Pune. And Abhishek, do drop in.
Thank you so much, ma'am.
You are welcome to
It
our
was
city.
an absolute wonderful session, ma'am. We learned so much from you.
Thank you. I know that this is this was capital of COVID a few days back, but Pune is Pune, so you are welcome anytime here.
Yes.
I'll see you soon, ma'am. And thank you so much.
Thank you.
Thank you.
Okay, everyone. Thank you.